All right, hi everybody. Welcome to our video walkthrough of DNA replication. So we'll start off at the beginning of the lab. I'm gonna go back and forth between Chrome and then also like what I'm showing you here. So let's start with Chrome. With DNA lab replication. I have this pulled up in smallpdf.com so I can modify it and then export it as a PDF pretty easily. You can also just do this in Word too. So if you wanted to pull up Word, you could do that. All right, so let's start off the bat. What three things make up a single nucleotide? Well, we know a phosphate group. We also know that there's a it says a single nucleotide, so we'll just say a ribose sugar, which can be deoxy or regular. And we know also a nitrogenous base. So the nitrogenous base is what is used to define what the phosphate group is, what the nucleotide is. I'm gonna try to move this up a little bit. There we go. Okay, so that's the three parts that make up a nucleotide. And then what type of sugar is used in DNA? Well, I answered right there in parentheses. In DNA though, so let's be specific, it's a deoxyribose sugar. The deoxyribose sugar. And then what is the name between all four nucleotides? Well, what is the same? Sorry, what is the same between all four nucleotides? What is different? Let's answer that question. The phosphate groups are all the same. Groups and deoxyribose sugars are the same. The nitrogenous bases are what's different. Let's see if I can move that up a little bit so I'm not blocking the text. Yeah, that's pretty close. All right, so the phosphate groups and the deoxyribose sugars are all the same. That's the backbone of DNA. And the nitrogenous bases are what's different. So next it says print out, and if I can stop sharing. Okay. So now it says print out and cut out the four nucleotides and color them in. So I had my 4-year-old color them for me. But what you can see, and I'll use my pen here, is I cut all the shapes out. We colored them. The phosphate groups I colored, they're like the H's. And I colored them in pink, or he colored them in pink. And then the sugars, the deoxyribose sugars, are colored in green. And then nitrogenous bases, I had them all do different colors. This is a G for guanine. And I joined them up like this, and you can do this too. So what you can see is that there's a pattern. There's a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, all down both backbones. So those are your two strands of DNA. They're oriented like this. And then in the middle are the nitrogenous bases. So you guys can pause your video right now and then cut them out. And then you're going to take a picture of this. So don't forget to do that. Okay, let's go back to our Google Chrome. So we did that, and we took a photo of the four nucleotides we constructed, and we used those. All right, and so now it said print out the figure on the next page. So I'm going to get rid of this. It says print out the figure on the next page. That's this guy. And label the nitrogenous bases with the initials of each base. So if you don't have a printer, you can just write phosphate sugar, like PS, 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 and then have the lines coming out to the bases. Okay. It helps to have a printer though, especially for online stuff. All right. And now we need to label the remaining pairs with an A, T, C, or G, and then answer these questions. So I'm going to jump back to this sheet. So we're going to stop sharing. And then back here. Okay, so we have our paper. So you've got thymine, adenine. Now what I want you to do is just fill in. I just want you to fill in what's left. And so we can see that this is a thymine. So I know this is a thymine. This is an adenine, so I know this is an adenine. All right, so now just to help us figure out what's going on in the middle here, it says thymine and cytosine are pyrimidines, so they have single rings. Guanine and adenine are purines, so they have these two carbon rings in their nitrogenous bases. Okay, and so we can see that the double rings are G and A. And so we already know what A is, so that means that these ones have to be G because they're a double ring, because they are a purine. Okay, and we always know that guanine bonds to cytosine, and cytosine is a single ring, it's a pyrimidine. So when we write a C there, when we write a C there. Okay. And so we know that cytosine and guanine are bonded by 3 double bonds, so we can see those in the middle. We can see adenine and thymine are joined by 2. All right, let's go back to our, I gotta sneeze again. All right, so we're back to our, so we labeled the 3 remaining pairs, and then we're gonna answer these questions. How does the orientation of the sugar group differ between the 2 strands? Well, if you remember, the strands are anti-parallel, and so you can see one sugar group is, you can see the oxygen, which is the O on that blue pentagon. That's the sugar ring, and so the oxygens are all facing up on the ones on the left, and they're all facing down on the ones on the right. And so the sugars are oriented in opposite directions. The sugars are oriented in opposite directions. They are anti-parallel. Oop, 2 L's, 1 L. Okay, to each other. Okay. And then it says, which part of the DNA monomer contains the information, like how to code a protein for DNA? Well, you might know this already, but it's the nitrogenous bases. This is because there are 4 of them, and they can arranged in many different sequences, whereas the sugar and phosphates are all the same between the nucleotides. So there's 4. Oh, you gotta pause this real quick so I can help Avery. All right, so it's the nitrogenous bases because they can be arranged in, I mean, there's four possibilities every single time you have a new base in DNA. And so that creates an infinite number of combinations. All right, what type of chemical bond connects the 2 complementary bases together on the strands? Well, you should know there's 2 of this type of bond between adenine and thymine, and then there's 3 of this bond between cytosine and guanine. So the bond is a hydrogen bond. And then it says, what type of chemical bond connects the phosphate group to the sugar and the sugar to the base as you go along a single strand? Well, let's think about that, right? So these bonds, I'm not sure what happened on my share screen. There it is. So these bonds between the sugar and the phosphates, these lines, what type of bond does that represent? Because that's a sharing of electrons. That's what makes this one molecule, is that all the electrons are being shared here. So let's see, that would be a covalent. That's what I would call a covalent bond, or covalent bonds, to be exact. All right, so we got those. Moving on. So now it says to cut the DNA strands. So we're going to cut strands. Oh, there we go. So we're going to cut these strands in half along the hydrogen bonds in the middle. That's the start of replication. So like the replication fork opens it up. Then we're going to write the nucleotide bases that will be added to form each new strand. So remember, they can only be added in a 5' to 3' direction. So one strand is going to be adding in one direction, the other strand will be adding in the other direction. And you will see what I'm talking about. And then we'll take a photo when we're finished. So don't forget to do that. I'm going to go back, stop sharing this, go back to this. And then what we're going to do is we're just going to cut right down the hydrogen bonds, right through the middle of them, like so. And you know what? It doesn't really matter. Let me just cut the paper in half. So now I'm going to take these and I'm going to put them on a new sheet of paper and I'm going to separate them. So this is like my replication fork. Now on the strand, like on the example, it should already have these, but there's going to be a thymine here. And this is the 5' end, because the way I know this is the 5' end is over here it says 3'. And so I know if I go across the DNA strand, I'm always going to be on the 5' end because the sugars should be facing the same direction. So the strand that I'm going to make is going to look identical to this one. And the one I make over here is going to look identical to this one over here. So we've got a thymine. The thymine is joined to the next nucleotide by covalent bonds to the phosphate to the sugar. And then cytosine is bonded to guanine. And so I'm actually replicating way, right orientation. This is how the place would build the new strands. Bind here. Okay. We've got a guanine, so I'm going to write cytosine here with 3 hydrogen bonds. Next sugar and phosphate, and then boom. And then I've got my adenine. So I've got thymine, adenine, guanine, cytosine, cytosine, guanine, adenine, thymine. And then look, I've got the 3' end of this. So now I've replicated one side of this DNA molecule. So that side's done. Now we need to do this other side. So this is the 5' end. That means this would be 3'. But I can't build 3' to 5', so I have to start up here. So this is going to be my thymine. And that's my 5' end. And now I can build down this way. And so this is what it's on. This is why in replication, you end up with, okay, helicase is opening the fork going this way. Or maybe it's opening the fork going that way. We could do it like that. So helicase is opening the fork, the replication fork going that way, separating these two colored sides from each other. Okay. We had an RNA primer here, polymerase 3 bound to it, and now it's just reading this template and it's copying the complementary side, anti-parallel, going in a 5' to 3' direction. And it would do that continuously, just along the whole strand as helicase unzips it. Now this would be the lagging strand, and it would have to go the opposite direction. So there'd be a primer up here at the top, there'd be a primer, and then the polymerase would be copying this direction. The problem is that the fork is going that way, so it's got to hop off and run ahead, and then copy backwards. Hop off, run ahead, and copy backwards. And hopefully that makes sense. That kind of goes hand in hand with the replication lecture we did. All right, so sugar phosphate. We got a cytosine here. And there's my guanine. And we know that guanine has 3 hydrogen bonds to cytosine. Sugar phosphate joining to the next one. And it's a G, so I'm going to put a C here, joined by 3 hydrogen bonds. And then finally, my last nucleotide here will be an A. And that'll be joined by 2 hydrogen bonds. And that will be the 3' end of that strand. And now if I was a polymerase 3, I would pick up and run to the replication fork, find my new RNA primer, and then copy back this direction. Okay, so now we're done with that. Make sure you take a picture of this because it's another submission right here. And I'll jump back over to my Chrome where I'm doing my PDF. Okay, so now we're on Chrome. And then now we can answer the following questions about DNA replication. And then we're done. That's it. So it's kind of a short lab. All right, I'm going to put this away. All right, during what part of the cell cycle does DNA replication occur? Well, we know that's during the synthesis or S phase, but also during interphase. So during interphase, there's a small segment called S phase, and that's when DNA replication occurs. Okay. How do your 2 completed molecules of DNA compare to one another? That's kind of a weird question. Sorry, this isn't my lab. I modified it, but I didn't change everything. So some of the questions read a little weird to me, but the 2 completed molecules, I assume it's talking about the ones we just did. and it said, how do they compare to one another? So here, type meth. Wait, what? There we go. Okay, they are anti-parallel. Well, two completed molecules. So they are identical. They're identical to each other, but one half is new and one half is old. So that's the semi-conservative model of replication. So basically the next question answers that. So it says semi-conservative. It means that completed strands will have one parent strand and one daughter strand in the same DNA polymer. I should say in the same double-stranded DNA polymer. That's semi-conservative. Conservative would mean that the two parent strands stay together and then they both get copied into the new daughter strands. But this one, the parent strands are separated and a new daughter strand is added onto each parent. That's the semi-conservative. DNA replication is currently going on in your body. Of course it is, dude. Yes. Yes, it is. It's going on in cells that need to repair themselves or parts of the body that are still growing. Also in the gonads. So like anytime you're making sperm and egg, you're going to go through DNA replication. Actually, just sperm, really. Eggs are already made. It's kind of weird. That's why there's like a biological clock for women, is that all of your eggs have, is that true? I'm trying to think. Well, I know that all the eggs are produced early on in development before you're even born. And then you have that set number of eggs. And I can't remember if they do go through replication before or after they're fertilized. I think there's something fascinating about, well, I remember learning about that as an undergrad, and I was like, wait, I know what it is. So meiosis, this is what's weird, is the second round of meiosis, right, where it actually becomes haploid, that happens after the egg is fertilized. And that's what I found was really weird. Okay, so anyways, DNA replication has already occurred in the gonads for females, but not in males. So the sperm will continually go through replication and division. Anyways, I digress. How's DNA related to chromosomes? Well, chromosomes are made of DNA and proteins. How do they compare? How's DNA related to genes? Genes are sequences, let's say they are specific sequences of nucleotides in the DNA polymer itself. I mean, there's specific sequence of nucleotides that code for a functional product, if we want to be exact, right? Compare human DNA to bacterial DNA. Since a human gene can be put in a bacteria and expressed into a protein, the structure of DNA in bacteria, well, this is kind of a given. They are the same. The difference between human and bacterial DNA is in the sequence of nucleotides. It has to be. But it could also be like the arrangement of the chromosomes, if you think about it, because bacteria have circular chromosomes and eukaryotes have linear. But that's not what it's talking about. It's talking about the sequence of nucleotides. That's what makes us different than bacteria. If the DNA sequence on one strand of DNA is GCCTAA, the complementary strand would be, well, just be what's bound to it. So G's bind to C's, C's bind to G's, so I got two of those, T's bind to A's, and A's bind to T's. Make a little song out of that if you wanted. And then boom, there you have it, you guys. And that's it. So make sure you can download this into a PDF form, and then you can upload your photos and attach them into the PDF, or you can upload them separate. It's all up to you guys. Hope this helped you guys out. I will catch you later.